Well, hello again. I'm Larry Smarr, Director of Cal IT2. A number of you will be over visiting us later today. For years, I've been telling FiRe attendees about the incredible importance of the microbiome, the most pervasive and important life on this planet, everywhere on this planet, including inside of all of you. But this time, we have a panel of people who really know what they're talking about with respect to the microbiome. These are three of our superstar faculty from University of California, San Diego, in the microbiome, and they are actually working together to create a major institute for microbiome research, but they're very diverse in their backgrounds, and we're going to try to paint a picture across the whole planet from starting inside of you or on you in the next half hour. So what I'm going to do is ask each of them to just give a quick station identification of where they come from at UC San Diego. And then we're going to delve into human health, into how we actually use the IT to understand the microbiome, into the environment and the planet itself. You want to start, Kim?
Sure. I'm Kim Prather. I'm a professor at Scripps Institution of Oceanography at UC San Diego, and I'm also the director of the National Science Foundation Center for Aerosol Impacts on Chemistry of the Environment. And I'll talk more about it, but basically we're very interested in how microbes in the ocean, how the ocean in general is working very hard to control the temperature of our planet and sanitary health. And so I'll focus on how microbes are working very, very hard in that regard.
Rob?
I'm Rob Knight. I direct the Center for Microbiome Innovation. That should be at UC San Diego, although it's true that it is to a large part based on information technology and understanding all that data. And I'm a professor of pediatrics, computer science and engineering, and bioengineering at UC San Diego.
And Jack Gilbert. I've just moved here from University of Chicago, so they managed to get me away from the cold into the warmth. But I'm now a professor of pediatrics and oceanography, which makes me the world's first pediatric oceanographer, or oceanographic pediatrician. But our research focuses mostly on trying to figure out how microbes communicate with each other and with the environment they find themselves in, whether that's in a human gut or in the ocean or in the soils. And we do a lot of work at the edge of translation, trying to get the research out of the lab and into small companies. And so I've launched 3 or 4 of those companies, and we are promoting the capability of intellectual property development with our own faculty, which is a major exciting drive for me, coming from industry.
So one of the things I wanted to start with, Rob, is we have a lot of people in the audience that come from information technology world, and you know, this world of microbes is largely invisible. And even if you look at it at a microscope, it doesn't help a whole lot. They're basically information creatures, and because of their DNA. And so I thought maybe you could just give us a little bit of how do you go from a sample, whether it's terrestrial or one of my 500 stool samples that you've got in your freezers,
That's a lot of crap.
to actually who's there and what their capabilities are.
Yeah, absolutely. Well, we're really creating a revolution here at UC San Diego in our ability to understand the microbial world, whether it's around us or in our own bodies. And you look old enough to remember what DVDs were. So each teaspoon of your stool has just in the DNA of its microbes the information it would take a ton of DVDs to encode. And a lot of what we're trying to do, so we get samples from anywhere, whether it's the human body, the ocean, the soil, even the sky, as Kim will tell you. And we extract the microbial DNA from it. And then we use high-end sequencing equipment like the Illumina DNA sequencing platform to read out huge numbers of fragments of DNA from the bacteria, the archaea, the viruses, the protists, anything else that was in the sample. And then the major challenge is to take all those fragments and piece them back into a piece of the puzzle back together so we can get a picture of what microbes were in each environment. But more than that, we're also integrating the chemistry, so we're using mass spectrometry to read out all the chemicals in those samples as well, so we can understand not just who's there, but what they're doing.
So just give us a scale of your lab here at UC San Diego. How many samples are you sequencing and then using computing to compute the microbes in every year?
Yeah, so we have capacity to process about 100,000 samples a year, and we've been working on a number of major cohorts like FinRisk, where, so one of the really exciting frontiers right now is using software not just to read out who's there at the moment, but to predict the future. So for example, from a sample of a kid 3 weeks after birth, we can predict how fat that kid is going to be at age 10, and we can predict how well they're going to perform on cognitive assays. What's perhaps even more amazing is things like the FinRisk cohort, where we have fecal samples that people donated to science back in 2012, back in 2002 rather, and now we have all their clinical records, so 17 years later, we can predict what happened to them afterwards, even things like all-cause mortality. So, you know, are we going to be some sort of microbiome gypsy where you cross our palm with silver and we'll tell you your future? Well, that would have sounded completely crazy 5 years ago, but the idea that from a technical perspective you could do it is looking much more plausible, and so then we start to wonder about the ethical perspective. But unlike your human genome that literally billions of dollars have been spent decoding, your microbiome is something you can change during your lifetime. And you heard yesterday from Sachin Panda about time-restricted feeding and the importance from Sachin and from Pam Taub about the importance of time-restricted feeding, and that it matters not just what you feed your microbiome, but when. The idea that you could take your microbiome and reshape it into a way that benefited your health, that's looking more and more plausible. And so a lot of what we need to do is we need to create a user interface for it, so it's not just a black box or brown box, I guess, but something that you can control and shape in the right direction.
And we're going to hear tomorrow from Mike Norman, who's the director of the San Diego Supercomputer Center, on the next panel I'm on. Why don't you just give them a scale of the kind of computing it takes to turn all of the sequenced information from the Illumina into actual taxonomic or metabolomic information?
Yeah, so the NovaSeq 6000, which is the latest platform we have at UC San Diego, you can do a 3-day run that will generate 20 billion DNA sequences, each of which is a few hundred bases long. So every run of that instrument is creating terabytes of data. Just in our lab, we have a few petabytes of storage, but also we use Comet, which, as you know, is a $26 million NSF-funded supercomputer at SDSC to do a lot of the processing for this stuff. And this really speaks to the ecosystem of what we have right here on the Mesa, right? So between the high-end computing, the high-end lab facilities, the ability to run large-scale environmental simulations, as you'll hear from Kim, it's really a confluence of different difficult high-end techniques that makes us make progress like no one's been able to make before.
And in about 60 or so, 50 or 60 you're going to actually get to go see that supercomputer. So just remember what it is that it's computing on. It's my poop.
And on that note.
So one of the things, Kim is an amazing scientist and just a force of nature. She's totally freaked me out. I know we all love being here in La Jolla by the ocean and seeing it's, you know, how healthy it is to be here next to the ocean, I thought. Kim, tell us about that.
You want the truth? The truth. Okay, you set me up.
These are truth seekers.
Yeah, so you know, we basically, you know, Rob talks about how microbes, as I say, influence human health. They're also trying to influence planetary health, and largely they're doing that through the ocean. And so a lot of people don't really think of the ocean. You think of the salty air you're breathing, right? And we have a huge facility that we've established here that allows us to isolate the ocean and the biology that happens in that ocean. They don't call it the living ocean for nothing. So when waves crash, they basically put bubbles down into the ocean, and you get an enrichment of bacteria, viruses, proteins. All of that is enriched in the air. And so one of the things we're learning is that it's actually a very selective transport process of what biology gets out. And then that biology, one of the things we showed was that microbes from as far away as Africa are changing the snowfall over California. So they've come all the way across. We find them, I fly through clouds. We sample what these clouds are made of. Turns out microbes are really good at making ice in clouds. They can form ice at relatively warm temperatures, which enhance precipitation. And so these microbes are definitely, they have a method to their madness, and we're trying to understand why only certain microbes get out, which ones get out and influence our weather, which ones get out and make it into our bodies. What is your microbiome, if you live near the ocean, shaped by these, and how do they get in? We're exposed to more air than anything else. And so living in a coastal region, for those of you from San Diego, one thing we hear a lot about is all of the crap that gets into the ocean, for example, from the Tijuana estuary. So millions of gallons of raw, untreated sewage are going into our ocean all the time. And we also have these huge pollution runoff events that are becoming more and more common with extreme weather events and flooding. And so you hear, when this happens, you hear, don't go swimming, don't go surfing. The oceanographers are always looking at how far up and down the coast these microbes, largely microbes with pathogens and toxins and other things. We hear don't swim, we hear don't surf, but we don't hear don't breathe. And so we are very focused, working with Rob and Jack and others, in what gets launched. What is in the air we breathe? It turns out that's what we understand the least about, but it's what we are here at UC San Diego uniquely equipped to go after.
And this is a real breakthrough. It really has just completely restructured our thinking all the way from weather to climate to health because of what happens when the microbes in the ocean are put into the air by the sea spray. So Jack, that's one kind of environment, but you are somebody who's studied a lot of different environments, including inside of our hospitals and other things, as well as on our farms, and how this impacts our long-range development of our immune system and things. Can you talk a little bit about some of those topics?
Yeah, Kim talks about what we breathe from the ocean, but you should think about what you're breathing in right now. Every single one of you is emitting around 38 million bacterial particles into your immediate vicinity every hour that you sit down in this room. Whether you're tapping on your phone or you're just sitting there breathing, you are literally emitting a little microbial cloud. Think Pigpen from the Peanuts cartoon.
Yeah.
And your neighbors are breathing that in, right? And it's stimulating their immune system. Now, if you were growing up on a farm, and we do a lot of work with people that do grow up on farms, you're actually breathing in a lot more than just your fellow humans' microbes from the environment in which you sit now. You're breathing in cow bacteria and viruses and things from the pig pen and the horses and the soil around them. We worked with the Amish communities and another community similar to the Amish called the Hutterites across the US to determine why the Amish have virtually no allergic disease at all in their population. There's no asthma, they don't really have any autoimmune disorders, and the Hutterites have these very elevated levels, right? They can have over a quarter of their population with asthma. The average in the US is about 8%. And we wanted to really understand that, and it turns out that it's what the Amish are breathing in. The microbes that we can isolate from their bedrooms, if we sneak into their bedrooms at night after they've gone to bed and suck up whatever the kids are breathing, when they breathe it deep into their lung during their REM sleep, right, they're breathing in the microbial world that they accumulated from the farm during the day. And that stimulates the immune cells in their lung and stops those immune cells from overreacting to the world around them. And when you sever that relationship, when you stop the children breathing in those things, it can have a significant effect on how their immune system develops, promoting asthma, promoting food allergies, promoting skin diseases. We've only just started to understand this phenomenon, but it's interesting. We found that if we expose mice and rats to the same kind of environment, we can actually promote the growth of bacteria in their intestine. It's kind of a weird jump there, but from what you breathe in, it affects how things can grow inside you. The bacteria in your intestine, if they get the right ones growing, can produce small chemicals which actually reshape many of the organs in your body and many of the cell processes. And that in itself can be a really quick way of taking those chemicals the bacteria produce, making them into a viable drug, and then adding that back into the system of young children to promote their immune system to develop quickly. So yes, we can live in an environment where there are no plants, there are no animals, there's no dogs here, there's no cows, but maybe we can take that farm, package it up in a drug, and reintroduce it into children that cannot get exposure to a farm. Or we could just take kids and rub their faces into the side of cows.
Which brings up
I don't know.
Which brings up your book. Tell us about dirt, why dirt is good.
Well, why dirt is good is because that's what the editors told us to call the book, right?
The publisher, yeah.
This is a book that Rob and Jack co-authored.
But the idea is that, the concept that we now live too much of a sterile life, right? Go back 100 years, 120 years, before the introduction of vaccines and antibiotics, our main predominant threat to our society was infectious disease. Thanks to vaccines, thanks to antibiotics, thanks to public health works, we've virtually eradicated that potential, but we've taken it a little bit too far. Now, for the vast majority of our time, we sterilize our hands all the time. We make sure our homes are bleached. I know some of our colleagues, no matter how much I tell them, use Lysol on virtually every surface of the home, right? And that actually is significantly decreasing the microbial exposure their children are getting, which seems like it could be good, right? Because we've been told for 100 years by every advertising agency that you've got to kill 99.9% of germs dead. It turns out that that's not the case, that the immune system needs that training, it needs that learning, right, from the microbial world around us.
It's actually a form of artificial intelligence or machine learning, that your immune system is training on the world around it, because after all, that's its job. It's got to protect you against that world. So if it doesn't know what the world is, how can it do that?
Right.
Exactly, and it's like we didn't learn the lessons from Silent Spring, and we're creating that situation in our own households and even inside our own bodies. And I think, Kim, you were saying just before this that even in the ocean, we're depleting those microbes with antibiotics.
Yeah, I mean, when we even put out treated wastewater, which we do all up and down the coast, we're releasing pharmaceuticals and all these sort of antibiotic drugs into the water.
Birth
and
control,
that's changing.
estrogen disrupting
cocaine, methamphetamines, all of that. And these are all hydrophobic things. They don't like water, and so we're getting all that into the air. But it's also reshaping the natural microbes that have been evolving for, over 3 billion years. And they have a method to their madness for this planet. And so, yeah, it's a big deal.
Yeah, and one thing that's absolutely shocking is just in the last 3 years, a number of groups around the world have discovered that a large fraction of the microbes in your gut, they're modified by drugs that aren't antibiotics but target those microbes nonetheless. And we know nothing about what those drugs do in the oceans and the soil and other environments that we're exposing to them.
So one of the things, Kim, is not a lot of these folks are professors, and I think they think of you as sitting at a desk and so forth, but you're kind of wild and crazy. I mean, tell them a little bit about just briefly about the plane rides you take into the heart of these weather systems to try to get the samples to let you understand how the microbes interact with the weather.
Yeah, so I am kind of crazy. In fact, we dropped 2 planes from the sky, almost to the ground twice, looking for these ice nuclei. So only 1 in a million particles. So at the center of every ice crystal, at the center of every cloud drop, is a particle, an aerosol particle, which is what I study. But microbes are the best, right? And so we fly through these clouds, and an instrument I developed early in my career, we sample the center of all the cloud drops and see what they're composed of and what those clouds are composed of. And then we look and see whether those rain or snow or do anything, nothing. In particular, as I say, we're very interested in the ice phase. Well, if you don't have something to form ice on, that water will stay supercooled liquid to negative 38 degrees C. So we pull that in, and if you give it an ice nucleus, it'll nucleate ice and magic will come out of that cloud. And so we're interested in how cloud seeding works, how these microbes work for cloud seeding. And so a couple times we've been flying very carefully through these super cooled liquid clouds looking for the magic ice crystals, and our plane quickly became the ice nucleus. And so we fell through from the sky, and word got back actually one time to our chancellor. I don't know if you know this story. And he called me and told me it was time to come home. So, I said, you just are worried about my funding. I bring in, but he was like, enough is enough. So I'm trying to think about ways now, after near-death experiences closer than I would ever want to come, how can we use like artificial intelligence And how can we use swarms of UAVs to go? My brain is the one that's telling the pilots where to fly. I have headsets on, I fly with the pilots, I tell them go back. That's not that great. But if we could train how we can get at the heart and understand what causes certain clouds to precipitate, we could actually control it much better through cloud seeding than we currently can. And Mother Nature does it really well.
Can I just add something to that?
Just in a couple of minutes, I'm going to call on questions. Of course, I can't see whether there's anybody at the microphone or not. But if you have a question, now would be a good time to get to the microphone. Yes,
The
Jack.
first patent I ever produced was to take those bacteria that nucleate ice and add them into ice cream. So they sent me down to Antarctica to find those bugs, not into clouds. And we find bugs that make ice cream taste smoother, which was the noblest thing I have ever done.
I'm really impressive.
Don't be saying that these professors aren't practical.
Ice cream's
So, Rob, I had another question for you. Your Center for Microbiome Innovation at UCSD is quite a phenomenon. I've been at UCSD 20 years, and in the last 5 years, these folks have just revolutionized our microbiome research to where we become essentially one of the key places in the world in microbiome research. But when the chancellor, you worked with the Dean of Engineering to come up with this Center for Microbiome Innovation, and one of the things is you reached out to our faculty to say who among all the different departments would want to work on microbes. How'd that work out?
It was amazing. So this was one of many agile centres that the Dean has put together to foster academic-industry relations, and we're by far the largest. So we have over 140 faculty members who are now members of the centre, including everyone sitting up here. We also have a dozen industry sponsors who are really supporting the research, both with an annual membership fee and with additional sponsored research that's making progress in things like the links between the microbiome and diet, and drug response, new technologies for reading out the microbiome, new ways of screening the impacts of industrial chemicals and household products on the microbiome, and so on. And that industry academic relationship, it's much easier to do here than it is at other institutions, and it's really allowing us to make progress that's not just intellectually interesting, but really matters in the world.
So one of the things I've noticed is in raising money, it used to always be just federal agencies and then, and of course, corporations we worked with, the private sector. But the role of individual donors and foundations and so forth, I mean, I, for instance, have now a foundation grant to study turning my transparent self that I did, some of you have seen, some of you will see later today, into ways that clinically can be used for other patients with Crohn's disease. What are you guys seeing as far as the role of foundations and individual giving?
Sorry, go ahead.
I was going to say, University of Chicago, I managed to raise $100 million from a single family who was interested in the microbiome immune system development. We currently are building a much bigger center, institutional structure here, and those visionary individuals are exceptionally important. But you talked about us being practical. I wish it were true that all professors were practical. A lot of the reason that we're not allowed to make that translational step is because the infrastructure does not exist. Our institutes become self-sustaining because we're actually able to take the intellectual property and revenuize it. We'll produce revenue from it. I'm a scientist, I can create words like revenues. Generating that revenue is incredibly important because it can feed back into the sustainability of our platforms. Right. And so we're looking for philanthropic investors, people who can actually see value in investing in early stage scientific endeavors that will actually pay off through translation.
Yep.
Yeah, there's questions out there.
Yep. So I'm going to try to see. Let's see.
Hi,
question
Larry.
over here.
Yes, Dave Dressler from National Space Society. When you consider what is dirt cheap, when you go to space, how cheap is dirt? and basically, you're starting with a sterile environment. Say you're setting up a greenhouse.
It's not so sterile anymore.
Yeah. How would you tune that biome to optimize, crop production, and also make sure that humans don't lose their natural immunity?
Yeah, so we're working with JPL on exactly that right now. So we've characterized a couple of dozen samples from the ISS so far, and we have an approved project to collect another 1,000 samples to build a very detailed 3D map of the entire ISS in terms
This is the International Space Station.
the International Space Station, the microbes and its chemistry.
It's covered.
It's covered.
Yeah.
Covered with human bacteria.
Covered with human
Ocean
bacteria,
microbes.
and it's
Ocean
completely
microbes.
isolated from the kinds of microbes that you would probably want to have interacting with your immune system over the longer term.
Let's
So
get
it's
another
certainly a
quick
very
question over here.
Not a lot of data.
So I wanted to ask a question about the ice nucleus. So you mentioned that, if I'm understanding this correctly, that the nucleus can be inorganic, maybe molecules, but also
Yep.
bacteria
Yep.
and live things.
Right.
So I assume maybe that's how, bacteria from the upper atmosphere gets down to us and how
Yep.
it goes from Africa to North
Absolutely.
America. How does inorganic versus live in ice nuclei, how does that change the characteristics of the ice?
That's a great question. So we're still trying to figure out what it is, right, about it. So there's snowmacks, which they use at ski resorts to make ice. In those, there's an ice nucleating protein basically the hand-waving explanation, because nobody's completely figured it out yet, is that you want something at the surface that templates water to be like ice, so that when it gets cold enough, it can basically pop.
It's like a fractal?
You want the hexagonal, sort of the structure of ice. And so it basically lines the water, so when it gets cold enough, it pops. And you only need one little tiny, it's not the whole particle, it's one little tiny spot, and that gets you over an activation barrier. The whole thing lights up. So understanding what we're trying to do is develop tools to get at what is that little magic spot. So getting at your question about, proteins are good. Inorganic. So the other thing that forms ice at relatively warm temperatures, not as warm as bacteria. Bacteria can make ice at temperatures as warm as -1. But dust. So we showed that dust from Africa is particularly good, and it's the mineralogy, and it's aluminosilicates. They're very hydrophobic. They also appear to be very good at templating ice. But that's, again, like -15, -20 degrees C.
Let's get another
Is
quick
that
question.
what happened in Iron Man when he went up? And
Yeah, absolutely. Yeah.
Another quick question over here.
Yeah, the thing about the microbiome is that it's fundamentally linear. There may be billions of these little guys, but there's only a few tens of thousands of species.
Yep.
So the dream is that this will be correlated very rapidly with your genome, your health, and your diet. And the combination of those four, probiotics will mean an entirely different thing, say within 5 years. Won't there be tailored probiotics?
Well, your genome explains about 2% of the variability, so that's not the main factor. Remember that a new microbe that actually does something isn't regulated as a probiotic, it's regulated as a drug. So if we isolated one from your body today, it would be on the market in 15 to 20 years, and it would take a couple of $1 billion to get through the clinical trials process. So what will happen much faster is what are called prebiotics, which essentially is fertilizers to encourage the growth of the bacteria that are already in your gut. And one important question is how much of that can come from diet. Another important question is which of the already approved food ingredients and drugs are out there that could have a huge impact on your health by modifying your microbiome, and we're just unaware of them.
Wait a minute, you're telling me, in effect, that there's going to be a black market of microbiota.
There is already.
Well, that's what we call probiotics.
A brown market, sorry.
Okay, we're out of time, but I hope that you get a chance to talk with our guests here.